Okay, should be good now. Sorry about that. My computer, the internet just disconnected, so I have to work around that. Okay. I hope this works. I'm going to start. Yeah, I'm going to start and see how this plays out. If not, I'll just be able to post it after the fact. But hopefully this works. I'm just going to ask, is it working? If it's working, can you guys write in chat? Could you guys write in the pop-out chat if it's working so I can know whether or not this is going smoothly at all? So if you guys, this is your first lecture, okay, cool. So if this is the first lecture that you guys have had with me, or first tutoring session that you guys have had with me, then I guess I'll explain who I am and kind of like the background before we jump right into things. I am a second semester 2X student. I've been tutoring. Awesome. That's dope. Okay. I'm a 2X student. I've been tutoring since the semester started for you guys. People who have come to my tutoring haven't, I guess, got to see me, know that I really enjoy this, and I thought that once the time was given to us, might as well take the opportunity to tutor you guys and see what I can do and see if we can get this to work in general. You guys have been very patient, and so hopefully this works out. I'll resist my best to say I have a tendency to say so I'll try not to do that. I have a way to make I have a way to get the sketchpad thing working. I think this works. I was told it works, so I'm going to use this when I have to draw things, and we'll go through this. Just before we get started, tips in general when we're going to start when you're going through this, because you guys might not know who I am or might not know, I guess, my philosophy on this whole program. I don't believe any of you. No, it's a sweeping declaration. I think that if you guys work hard enough, you can make it through this process. I just think that a lot of other things get in the way in terms of our own, whether or not we don't, how do I word this? Sometimes our pride gets in the way a lot, and we don't take tests that we know we're going to do poorly on. We don't put ourselves in positions where we might find out we don't know things so well, and we refuse to find out where we're not so good at things, and that gets in the way of us. That gets in the way of us being the best that we can here. I'm going to say that if you have any questions, if there's anything that pops up, write it in the chat so that I can work with you guys. It's really weird. It's a really weird process for me. I've never tried this before. I've never been tutoring to people who don't get to who I don't get to see. Sorry, I had a message, so I got distracted. Should probably avoid that. But I never tutor to anyone who I don't get to see the reaction and get to see whether or not I'm hitting the mark with my explanations. But if you guys could let me know in chat when we go through some stuff, if things work, if there's any questions, if it's not making sense, and then that'll help me get through this process. Okay, great. Also, we have quite a bit of time together. At least I've allocated a couple hours to this, so there's no need to rush through things. I'll take my time. I'll try to explain things as best I can, and then maybe I'll have another session where we can go through questions if that's helpful. So when it comes to the mutations lecture, I know biochem is a big issue for a lot of us. I know first semester that was a tricky one, and it's taught by professors who are like geniuses and who don't do necessarily the best job of making sure you get it. But what you have to really do in biochem is kind of look at the bigger picture of the lectures, understand the lecture in its entirety before you start working on the details. Sometimes we get so fixated on making sure that we know every detail that we forget to actually know. We forget to see what it is we're learning in general. It seems intuitive, but it happens all the time. You just like so zoomed in every lecture, trying to learn all the details on the slide, this slide, next slide, next slide. Then next thing you know, you have no idea how these things go together. So in the mutations lecture, there's a distinction to make. I'll try to say this clearly and hope that you guys understand it. Sometimes they are giving you So they're going to describe 5 different mutation repair mechanisms. There's 5 of them, I believe, 5 different mutation repair mechanisms, and then a bunch of examples or clinical correlates that associate with these repair mechanisms. Some of the pathologies that they reference are fixed by the repair mechanisms. So some are fixed. Other ones manifest themselves when the repair mechanism is not working. And the distinction you have to make. So basically all you got to know in this lecture really is the 5 different repair mechanisms. That includes the enzymes in the stepwise fashion of every mechanism, and then the mutations, the mutations that can be fixed by them or that manifest themselves as a result of that technique, of that repair mechanism not working. I hope that's clear. If it is clear, always use the chat for me, guys, because I have no way of knowing anything without the chat. The chat helps me know that we're going at a reasonable pace, that you guys understand the distinction that I'm making, that it's sounding good, it's flowing, because I don't have anything else. I can't do a giant Skype call. I don't see it as a viable option. Besides, there'd be like 30 faces at the bottom, and I don't think that works. So thank you, I appreciate that. Okay, let's start. So I'm going to go into this and say, okay, so the first type of repair mechanism, I have a metaphor for it. It's helped students in the past. I'm going to use it now, and I hope that it's clear. When I go through this sketchpad, I'm not able to see the chat, so I'll be drawing, and then I'll go back to the chat and see if it makes sense. But still feel free to write anything, and I will check it as soon as I'm done this. So the first one, I call it a fallen tooth. So if you imagine DNA as a set of teeth, okay? There are certain mutations that can occur that cause one tooth to fall out. So how do you erase really quickly? Yes, that cause one tooth to fall out. There. So this tooth is gone. Now, there is a repair mechanism that is used to fix this issue. And these, when in your slides, this will be referred to as an apurinic site. Apurinic site. So you know how there's purines and pyrimidines? I don't know what it would be called if a pyrimidine falls out, but we were only taught it if a purine falls out. Maybe purines are more inclined to fall out during mutations than pyrimidines, so apurinic sites are more common than pyrimidine, than the lack of a pyrimidine. So they call it AP sites or apurinic sites. So it's A meaning lacking and purinic meaning purine. So apurine sites are sites in which a purine has fallen. So they have references. I'll go to the slides after to help reinforce it, but there are situations in which a mutation can occur that causes the base to fall off. And in the event of the base falling off, you need to repair it. So there is 1, 2, 3-step enzyme process that repairs it. The first thing is you have to, because if a tooth falls out, imagine that the tooth falls out, but the backbone, this right here, is still present. And if it's still present, then you cannot insert a new tooth because the tooth comes with its own root. So you have to remove the existing root and then insert the tooth in its entirety. So you have a situation. Oh, that's kind of cool. You have a situation where first thing you do is you remove the backbone. Okay, and this is pretty simple. The name is AP, because we said AP is apurinic, endonuclease. Endonuclease, endo meaning in the middle, exo meaning on the outside. So endo means it's from within, as is the case right here, you can see. And nuclease is cleaving. So that's self-explanatory. Now that we have cleaved the backbone, we've removed the backbone, we've cleaned up the site. Basically, we came in with a fallen tooth, we removed the gums, or we removed the root. So now it's time for the insertion process. So the second step I should color code these, that would be interesting. Second step is DNA polymerase beta. I'll have to go on a side note, but you have to remember all the polymerases. You have to know that alpha, beta, delta, epsilon. There's 4 of them: alpha, beta, delta, epsilon. Yeah, and you have to know each one's function. And each one has a different function. I'm going to say them. If you're following me in my notes, you should be able to write them down, and then I'll go check and see if this has all been smooth so far for you guys, because I can't see the chat, so I'm literally just I'm explaining to myself conceptually. Alpha puts on primers, so alpha's primers. Beta is for repair. Delta is the lagging strand. If you think that the delta symbol kind of looks like a G in a weird way, backwards G, upside down G, kind of looks like a G. There's a lot of G's in lagging. Delta's lagging, epsilon is leading, and E is in leading. There's no E in lagging. So epsilon leading, delta lagging, beta repair, alpha primers. Done. Now, that's a little tangent, but you need to remember that. It's not that big of a deal, guys. You write it in a sticky note, stick it on your mirror. I tell people this. Don't complain when you have to remember certain things. I'll do my best to help you conceptualize, but there are things that you'll just have to remember. That's the nature of this whole thing. So here we are. We've added this using DNA polymerase, but you can see that there's a step missing. I don't know about step missing, but there is a next step that you can conceptualize without really even knowing the actual technical process. You know that in order for it to be properly integrated, you have to connect it to this and this. So this part needs to be dealt with because they're loose. It's a little phallic. Undo. And how do you know what you already know of an enzyme that cleaves, that anneals loose ends? And if you're following along, it's DNA ligase. So those are the 3 enzymes that would repair a fallen tooth. Okay? All right. I'm gonna go check and see how that was for you guys. How's that going? I'm back to the slide. I'm back to you. I guess you guys know where I'm back. Is that okay so far? Anyway, I don't know if there's probably obviously a delay, so I kind of have to just continue and then read when you guys post. But please tell me if it's not making sense or if it is making sense. I know I don't know how to pronounce your name, but I'm not really good at name pronunciations, and I don't want to butcher anything. So I'm just gonna say thank you. Okay, so these we'll get there. Types of mutations. You guys should know this. This isn't anything. This isn't anything that you should have any trouble with. This is conceptual. You probably learned this in undergrad. If you haven't learned it in undergrad, it's just things to memorize. Single base means one thing changes. Insertion and deletion means you can have things inserted or deleted. It's not too complex. I hope that you guys haven't had an issue with this. But now, this is also missense, nonsense, frame shift. You know, if you add or insert, if you insert 3 bases or remove 3 bases, it isn't a frame shift. Just something to make sure you guys are aware of, because you know that every 3 equals 1 codon. If you have a strand and you insert 3 right here, you don't actually shift the reading frame at all. It still reads, it just adds 1 base. So you're still like 1, 2, 3. These are amino acids, right? You're going to eventually get to here, and then you're just going to add 1 amino acid and then make another. So this is, so what I'm trying to say is insertion of 3n bases does not equal frame shift. I hope that you can appreciate that makes sense intuitively. If you add any multiple of 3, then you're just adding bases. You're adding nucleotides that will then directly add a codon. or an amino acid. So you're not actually shifting the frame at all. Just take that in. We'll continue. Now the second metaphor, the second example. So we said one was fallen tooth, right? Fallen tooth, and we're gonna go with done. Okay, second one is a rotten tooth. It's a little different, okay? Now a rotten tooth. So you know that every base has to match with the base below it, right? That's the idea. That's why you can use those laws. I forget what they're called. Kirchhoff laws. You can use. I don't have to remember this stuff. You can use laws that help you know that if you know one strand, you know automatically the DNA composition of the other strand, so you know the total composition of A, T, C's, and G's because they match. So in the event that you have a mutation that causes one to be messed up, so this is my example. That's a rotten tooth. Now, a rotten tooth is a little slightly different, but not much different than a fallen tooth. Imagine a tooth that eventually rots so much it falls off. So conceptually, it's pretty similar. There is just one extra step that you have to go through. So if I were to ask you without knowing anything, how would you get from situation 2, which we've described, to situation 1? How would you get from how would we make this situation like the one that we had just learned? Grab some tea. Sorry. I do have a sore throat. I woke up today sick, so it's like a test. Do you really want to help these people? Now you're sick. Decide. So if you had a rotten tooth, and I told you how do you make it look like that, you would say, well, okay, based on my limited understanding of anything, I would say just get rid of it. Right? Now that looks like the first one that we did. And you would be absolutely right. So there's not much there. And I believe the enzyme that does that is DNA glycosylase. I have to check that, but I believe that's the case. Now, we've removed the rotten tooth. And like we said, a rotten tooth is when you have one base changed to something else. So when you have, for example, you have an A changing to a C. Okay? Now we have a site that it's the same as an apurinic site. There's nothing here anymore because it falls off using this enzyme. I hope that makes sense to you. So after you remove the rotten tooth, you then you have exactly the same position as the first situation. So what did we say the list of enzymes were? We said after that we had to remove the backbone. This needs to go, which we said was done by AP endonuclease. And then after that, we said that we've removed the backbone. Now we can start repairing because we've cleaned up the area. The thing is, I can't delete without deleting the whole line, but it's okay. So I can just go like this. Now we've got to this situation using this. Now we have, let's use a different color, 3. We said after AP endonuclease, we start repairing. What did we say was for repair? DNA polymerase. What is this? You guys should be following along. Repair is beta. I hope that you guys remember. Again, I'm going to go this one more time. This is what I do, by the way, when I write my notes, when I review. Every time you get to a situation where it calls into question another set of information that you know, go reinforce that other piece of information. Don't just learn repair is DNA polymerase beta. Or in this step, in this mechanism, I have to use beta to put in the new backbone and base. You connect all your information. That's what gets people in this whole process, is that, like I said at the beginning, you spend so much time learning slide by slide, fact by fact. You're not reinforcing, you're not connecting. You have to do that. They're not going to do that for you. And so don't cheap out. Don't take the easy route in the sense of just like learning each piece of information. Like, oh yeah, I know that this reminds me of something else, but don't worry about it. Right now I'm focusing on this. No, go back. So we said alpha was primer. Beta was repair. That's easy. Now, how do we distinguish between delta and epsilon? I'm pointing at them like you can see my finger pointing. It's hard to teach. How do we distinguish between delta and epsilon? Epsilon is like E. E is in leading, so you know this one is this one's leading, this one is lagging. Delta is lagging. So one more time, lagging. We gotta make sure that by the end of this should be cemented. I hope so. Okay. So that's that. Okay. Now back to our last step. So we've done this, and we said there's one more thing we got to do. And if you guys are following along, then you know that the last step is ligase. So that just boom, boom. That's what these things are right here. Done. So, do you see the connection between a fallen tooth and a rotten tooth? Fallen tooth is an apurinic site, a site that has no purine in it, so the tooth has fallen out. Once the tooth has fallen out, you just got to remove the root. You remove the root, then you insert the whole new tooth with the root, and then you, I don't know, put braces on it, if metaphor needs to continue. Then you ligase the tooth in, and now it's integrated into the rest of your mouth. Great. Rotten tooth, the tooth is funky. So you get in there, you see a funky tooth. First thing you do is you got to remove the tooth. Then once you remove the tooth, then you got to remove the root. It's the same idea. Or you can think, you have to remove the tooth, which includes the root, which doesn't come as one step. First you remove the tooth, then you remove the root, then you add the new tooth, then you integrate it into the rest of the mouth. It's exactly the same with one extra step. Now that you fully understand the mechanism, I'll check that assumption. Do you guys... Perfect. You guys following along? I'm back to the chat, as you can obviously tell. I hope that makes sense. Those are two of the mechanisms so far. Okay. You guys have sank that in. There is great, guys. I'm glad to hear. At the end of the day, I am doing this because I enjoy having you guys have a better understanding of the process. I've always loved teaching. I think it's fun, and it's hard work, but it's good. It's enjoyable. So now I'm going to go through the slides and see what we're referring to now. This so far is pretty intuitive. I will help you guys go through this. Would you guys like me to go through the difference between gain of function, loss of function, haploinsufficiency, haplosufficiency? Would you guys like me to take the time to explain that? Do you guys have the distinction between the 4 different types? Do you guys understand this? What's going on right here? This jazz. Go over it? Okay. I guess I'll just... Okay, I'll go, okay, no problem. I'll go over it. So I'll explain the difference between, okay, yeah, that's reasonable. We'll go through the repair mechanisms, then we'll come back to it. Alright, that works with me. That's perfect. Okay, so I'm just trying to, whoa, what happened here? Okay, so these are all different ways that things can go wrong, right? And I told you that as things go wrong, there are different repair mechanisms to fix them. So these are the ways that things go wrong, and it's your job, because I'm not going to do it for you. I'm not doing everything, right? I'm just trying to help you guys get the bigger picture, and I told you at the very beginning, what you need to do is understand the 5 different ways of repair, and then know the distinction between some being repaired by this, and some mutations that they describe, some disease. Like they're going to talk about ataxia, telangia, ectasia. You guys remember that? You don't really know it. You just know it's a thing. What it is a manifestation of the inability to repair double-stranded breaks. So if you have a double-stranded break and you cannot fix it because that mechanism isn't working, then they accumulate and they manifest itself as the disease known as ataxia telangiectasia. So that's a distinction, as opposed to like pyrimidine dimers, which happen due to the sun, which can be fixed using one of the mechanisms we're going to go through, or like if you have deamination, or so these are all different. These are all different repairs, different mutations that are then fixed in different ways. Okay. So if I show you when we start, so if I show you the best way to explain. So yeah, these are the five. These are the five. Well, I guess there's six. Not really. There's five, because 3' exon nuclease activity isn't really, that's while you're actually making it. So imagine you're putting new bases, and when the bases get put wrong, you can immediately fix them because your polymerases have nuclease activities, exon nuclease activities. So that's different than a set issue in the DNA that's lifetime repair of issues. But we saw AP endonuclease is baseless or apurinic sites. Apurinic, AP equals apurinic, so lacking a purine. That's pretty simple. I feel like there's a slide that's super informative. If I can show you, just can't seem to find it. I don't know if you guys know the slide I'm talking about, but it's the one that breaks down the process. Sorry, give me a second, guys. Here it is. Okay. So, apurinic site versus base excision repair. If I'm going to, we said what type of repair was this? This was one fallen tooth. I hope you guys can see that. This isn't too small to you guys, right? Okay, this is okay. I hope it's 15. So the first one we said was fallen tooth, and the second one we said was rotten tooth. So now that you understand it conceptually, all you got to do is remember the name of it. So it's not that hard once you. That's the thing, is you don't want to spend time memorizing all the names of things you don't understand yet. So imagine, I can imagine the process with which you guys go through things. You'll go, okay, thing I don't know, named by something I don't know, does something I don't know. So that's hard. It's a lot of things you don't know. What you want to do is understand the concept. Understand the concept first, understand the bigger picture, know the mechanism, really get that, whatever that means. Sometimes it's understand the concept, sometimes it's know the mechanism. It's look at the grander picture. Once you get that, then just memorizing a name isn't so tricky. Okay, so fallen tooth, apurinic site. Rotten tooth, base excision repair. Think of that conceptually, base excision repair, meaning the base is still present. So obviously it is the rotten tooth situation because the base is still there and it needs to be repaired, as opposed to an apurinic site where there is no tooth. So the site is apurinic, lacking purine, and then you go through the process. So here they gave one for you, deamination. If they gave you a deamination mutation, now you know deamination is a mutation, is a problem that can be fixed using base excision repair. Why? Because a C got turned to a U, and then, okay, that's a messed up tooth. What's the first thing that needs to happen? You remove the tooth. There, now we have a backbone with no tooth. Now, we see AP endonuclease, polymerase beta, then DNA ligase, and we're done. Do you see the exact mechanism? It's one extra step between the rotten tooth and the fallen tooth. Here's the fallen tooth. There, it's already here. And now you know another example. Depurination is depurination. Like the name is it's intuitive. So deamination versus depurination, they're both different types of mutations that can be that can be repaired by different types of mechanisms. Does that make sense? Good? Okay. Let me go through the third one now. So, third one. I'm going to continue. I hope that makes sense with you guys. Okay, so the third one. So the first one we said, and this is how I also do it. You see how I don't just write number 3 and continue? I'm not doing it just because for tutoring's sake. This is how I write my notes. So I would go fallen tooth. When I go through the process, I'd go fallen tooth and rotten tooth. You know the names now, so if you want to put next to the name, apurinic apyurination sites, or AP repair, and this one you want to put base excision repair. That's base excision repair. I hope that you followed that. Now, so when I go through my notes, I go, okay, done. I've explained the first one, I get the second one, now we're on to the third one. So every time I write my new one, I don't just, I keep myself going through the process. I don't lose sight of the bigger picture. Okay, there's two done, there's another one. Here's the third one. It says nucleotide excision repair. Now, my metaphor kind of gets thrown out the window here. I helped you with the two teeth, 1 and 2. The third one, you can kind of think of it like, I don't know, like braces, I guess. But basically, I'm gonna start calling this NER, by the way, because why write the whole thing out? If you're following along, NER is NER. Great. Now, in the event, we have situation, bunch of different bases. All right. I don't know why I'm drawing ends. It's such a bad habit. Okay. Now, in the event, let's say these three are messed up. And if you can think of an example off the top of your head where even two are messed up, let's make it simpler, because maybe you'll be better at thinking of an example. If I can give you an example, give me an example where you can have a problem with two bases next to each other. And if you're following along, maybe I'll just say, maybe the question will just say sunlight. You know, and you go, sunlight? What kind of mutation is sunlight? And then you have to think, pyrimidine dimers. That's one example. Remember how we said this lecture, don't lose sight of the bigger picture. This lecture has different mutation repair mechanisms, some mutations that can be repaired by these mechanisms, and some mutations that manifest themselves as a result of a malfunction in the repair mechanisms. Does that make sense? Don't lose sight of that. That's why this lecture can be a little confusing. Some of these, it's not that Cockayne syndrome or xeroderma pigmentosa is repaired by this mechanism. This mechanism not working, by this I mean this mechanism not working, NER not working, results in Cockayne syndrome or xeroderma pigmentosa, depending on where the NER isn't working. Say that again, because it's fundamental, but like it's fundamental to understanding this lecture. I don't mean it's fundamental like you should all know it, but you should all know it eventually. The pyrimidine dimers are an example of a mutation that is repaired by NER. In the event NER has lost its ability to repair, there are some mutations, there are some diseases or pathologies that manifest themselves that have specific names that are a result of NER not working. The two that you need to know are Cockayne and xeroderma, and they are two different syndromes depending on where NER isn't working. If NER isn't working in the whole genome, it's not only not working during transcription. If it's not working all the time, then you're left with a very severe, very extreme xeroderma pigmentosa, because there's no way of repairing it. There's no way of repairing any sort of issue where there are nucleotides attached together, wrongly synthesized, any issues that involve more than one nucleotide. There's no way of repairing it, so it manifests itself as xeroderma pigmentosa, as opposed to if it's just messed up during transcription coupled. I think C is coupled, and connect that with Cockayne. Transcription coupled Cockayne. Transcription coupled Cockayne. So that'll help you remember when. If NER is not working during transcription, coupled means just attached to. So if transcription coupled repair, or if NER isn't working during transcription, then you get Cockayne syndrome. If it's not working in the entire genome at all times, then you get xeroderma pigmentosa. Does that make sense? Great. I mean, you haven't wrote anything yet, but I'll take the safe assumption that I'm going at a pace that makes sense to you guys. And do you see here 2 types of repair? And genome-wide, xeroderma. Transcription coupled, cockane. This is different than, these aren't things that are fixed by this. These are things that are resulting because the thing that fixes it isn't fixing. Okay, great. I'm happy to hear that, guys. Okay, we're gonna move on. Oh, no, we're not. Yeah, no, we're not. Sorry. Where was I? Let's get this out of the way. Okay, so we have this pyrimidine dimer example. Great, you guys are following that. I saw some of you writing that in chat. That's dope. Okay. Now, you have to, now remember, there's a couple factors. These are connected. Okay, these are H bonds. Okay, you know that. When you have one messed up base, it was different. In the example of, we said that, let's say the rotten tooth, right? In the example of the rotten tooth, it's safe to assume that this connection, something's messed up here because it's not in the correct spot. It's not the correct base anymore, so they're not going to line up perfectly with this one. So when you cut here, it was easy to just remove because there was nothing holding it in. Now in this case, it's a little different. So first thing you had to do, and now I got to remember the enzyme that does it, which I don't think I have off the top of my head, but I can simply look it up. First thing you have to do is cut here. Separate it from the rest of its teeth. So make it stand apart alone. Make it stand apart alone or separate it from the rest of the strand. Now that it's separated, it doesn't just fall off like this one did, where you could just cleave this off and then it goes. Because of these right here. This is preventing it. So now what do we already know? Let me say the first step was this one, right? This is 1. So let me go check the enzyme because I don't have it off the top of my head. Let me go check the enzyme. Nuclease. That makes it easy. See this right here? Nuclease. So let me go back to the thing. Here we are. So the first thing, nuclease. Okay, that did this section right here. Well, if you're following along, then you're following along. That did what that did. Now that did what that did. Incredibly informative words. Now the second thing, let me give you a different color, is if you guys are cheating on, or paying attention or cheating, both ways. Sometimes it's the same. If you're paying attention, then you would have saw the next step. Like I said, we've solved the upper issue. We've solved this issue. I don't mean to confuse you with the color code, so I won't. But we've solved that issue. But we haven't solved this issue. What do you know breaks H bonds to help separate one strand from another strand? Just think about this conceptually. If you're following along, which I hope that you are, helicase. Because you can't just rip it apart. Like, you can't just take it off. You have to remove the bonds that connect it to everything. It has to be standing in isolation, and then it can be removed. So now that we have done that, we're left with, I'm gonna delete a lot here. Okay, so if you're following along, oh, oof, that was not deleting. Here we are left with something that looks like this. Oh, no, we're not. So once the helicase has been functioning, now it's safe to assume that with nuclease and helicase together, it's gone. Now this looks like the fallen tooth, just a bit bigger. The process is still the same. If I were to ask you what you would do, you'd go, okay, let me put on these 2 teeth. That was done by DNA polymerase. Now which one again? You guys are like, oh, beta, it's beta, dope. I have to pretend like you're super following. And then again, I would exactly the same way I would do this. I would go, let's see if I remember them. Alpha, beta, delta, epsilon. What are the 4? DNA polymerase alpha, you guys know it. Beta, you know it. Delta, you know it. Epsilon, you know it. Done. You can go, okay, great. I'm actually starting to separate those 4 different types. And that's what I mean. Every time you write DNA beta, you now instantly should remember the other 4 or the other 3. and that helps you cement things. Speaking of cement things, tutoring transition. Next step is this. And we've already done this process, guys. This isn't new information. You understand this. Don't memorize it, understand it, and then realize that, oh, I don't actually have to memorize that much. And that does this. Great. Okay? Does that make sense? So really, nuclease, helicase, and then the same process over again. Oh. Yeah. I'm going to finish off this process. Now, the last thing, or not really, but we said NER, the last thing. What an overstatement. There are two types. Let's say two types. There's transcription coupled NER, and there is genome-wide NER. And I'll tell you, when I see genome-wide, I think of also GWAS, genome-wide association studies. So maybe when you see genome-wide, you can go, genome-wide, I've heard that before. What's a GWAS again? And then you go back and go, oh, GWAS studies. When do I use GWASs? Why would I use a GWAS study? What's unique about a GWAS study? While you're at it, why not? Don't be lazy, basically. Find out, oh shit, I don't remember GWAS studies. Then go back and learn them. Like it's could be worse. Now, if just in general, guys, it is always better to voluntarily be an idiot on your own accord. Find out where you don't know, find out the limits of your knowledge, go push them, go put yourself in positions where you don't understand things, as opposed to avoiding the things you don't understand in the hopes that they won't exist. You're still going to get to a test, you're still going to be confronted by it, but now you haven't got yourself in order. You haven't put yourself in order so that when the problem comes, you can rise above it. And the only way you can do that is by voluntarily confronting the problem of your own accord. And that applies to everything in life. At least I believe so. I'm not here to give you life advice, but it's functional for med school, if not anything else. I personally try to approach that with everything that I do, but to each their own. So, back where I was. If tcNER is not working, transcription coupled NER is not working. So let's go with no, that doesn't make sense. That doesn't equal. Let's say not working. Then you get coupled, what did I say? Coupled, see, you're going through the process. Coupled Cockayne syndrome. Cockayne. If genome-wide NER is not working, very low, boom, dramatic, then you get xeroderma pigmentosa, blah, 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 you know the word. Also, another way to genome-wide. Genome is everything. If that's not working, it's safe to assume that 0 is a result. Genome-wide, xeroderma. I kind of think of this as no skin. Xeroderma, 0 skin. I know 0 isn't necessarily 0, but 0 skin, genome-wide. If the whole genome, if all NER is not working and in the entire genome, it's safe to assume that it would be pretty serious. So, xeroderma. Okay? And those are results of them not working. Do you see the distinction? You do see the distinction. You guys know the distinction. That makes sense. Great. Perfect. Awesome. Okay. Great. Next. I'm glad you guys are following along. I actually am curious as to how many of you there are here, but it's probably my own issue. I should probably not care. Great. Now, another repair. Double-stranded brace. I think there's one other one that they post-replication mismatch. Yeah, exactly. Okay. All right. So I'm going to go through this now. It's actually it's been 50 minutes. Do you guys want to take a break? Do you guys want to finish the 5 repair mechanisms and then take a break? So break now. Thank you. Okay. All right. Okay, cool, guys. Obviously, if you want to take a break, I'll say I'm gonna give 2 minutes or a minute to go take a pee or something. If any of you have to go take a pee, um, 1 second, guys. Presley Goodfella. That's a dope ass last name. Um, no, I don't need a break. But I am going to take. I'm going to take a minute or two. Let anybody who wants to go take a pee go take a pee, because you can't learn on a, if you're thinking about peeing. It's just those are just facts. Um, oh, it's pretty good. It's not bad. It's not bad. It is not bad. Okay. I'm going to use the actual words now. So far, that's what you've learned. That's what we went through. And so you go, boom, I get it. Well, first you go, okay, follow them all. And that's it. And that's kind of how I go through my notes. I always rewrite the list, and then I check mark them as I still understand them, and then I get to go through the next one. Okay, I'm going to start now. Okay, cool. Now, where is this process? So I just gotta make sure. People have asked to join. I hope anyone who is trying to be on this is on this. That's all I can hope for. Okay, great. So post-replication mismatch repair. This one's pretty easy. Because it's very familiar. Post replication. So after replicating, mismatch repair. Post replication mismatch repair. In the event that you have a strand, and you have another strand, and at one point you have a mismatch, so they don't bind properly, you will then produce a bump in the rest of it. If you have a mismatch, I'm going to write MM. If you have a mismatch, then you will get a nick. I think that's one that. Now that means that everything is messed up from here to here. So there's a set of proteins, and that's what makes this easy is that the protein is called DNA mismatch repair proteins. What they do is they do some weird folding thing. I'm not going to even begin to attempt to draw it. That leaves you with this. It leaves you with a gap from mismatch, where it used to be, to nick. So this part goes away. This section right here goes away. Okay? And then, after we've done that, now it's exactly what you've already seen. This shouldn't be tricky. You got to add the new ones and then seal them. So we said there's DNA polymerase. Which one? Beta. You guys remember this? And again, you go, oh, beta. I'm drilling it home, but it's literally what I do every time I write it. So at least I'll remember until I no longer need to write it at all. And then every once in a while you'll see it again and go, ah, do I still remember it? When I was tutoring with you guys and I first had to write DNA polymerase beta, And I was like, oh, let's see if I still remember it. Alpha, beta, delta, epsilon. And then I got to write all 4 and go boom, 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 boom. And it was done. And I realized I still remembered it, and it was functional. So I'm telling you, it's the same idea. Now, after you have that, you're left with this. Okay, now there's one last step. You guys know it. DNA ligase. Done. Okay? Simple enough. Simple enough. This you've seen a bazillion times. Bazillion's an overstatement. 3 times. 3 times? 1, 2, 3, 4. You've seen it 4 times. It's in every one. It's in every one. So you've seen it every time I've talked about it. So this is a standard. All you got to know is the things that are extra between these. So in this one, we said the extra one was AP endonuclease. This is something you should be able to do, right? AP endonuclease. In this one, we said that we had to remove the base first. So that was DNA glycosylase, and then AP endonuclease. This one we said was we have to cut off the ends, nuclease, and helicase. And after that, in all those examples, we're left with something that looks like this, with something missing. Whether it be a single base or a bunch of bases, then you do this every time. That makes sense, I hope. Yeah, so it's called DNA mismatch repair proteins. And this is that crazy thing that I didn't attempt to draw. And this is from mismatch to nick. And then it's left with a hole from mismatch to nick. And then you use polymerase. And see how I write it in my notes? Beta and ligase. Great. So that's the 3rd or 4th one. So you look at the list. We've covered this one. Using mismatch repair proteins, we covered this one, we covered this one, we covered this one. All we have to do is this one. That's the last one. And so we're going to carry that into our last one. This makes sense. I'm going to get rid of it. So we said again, 1, 2, 3, 4, 5. We said this was AP. This was base excision repair. And this is how I write it after. You don't have to write all the details. If you start understanding it, nucleotide excision repair. This one is DNA, it's post-replication. Post-replication mismatch repair. I know mismatch is one word, but MMR, I don't know. PRMMR. And the last one is double-stranded. I usually start it like this because I like double-stranded, like you'd write double-stranded DNA. Double-stranded break. Double-stranded breaks. Okay, so we have covered this. We're on to the last one. Now, in the event that there's a double-stranded break, it's very simple. It means both strands break. Okay, let's say these are sealed off so we don't get confused. So here, right here, is the double-stranded break. There are 2 routes that it can take. Imagine a bridge breaks. Okay, if a bridge breaks, try to conceptualize this for you. If a bridge breaks, is obviously missing pieces. Now there is, let's say, a shitty repair guy and a good repair guy. I apologize if I swear. There's a poor repair guy and a good repair guy. A poor repair guy will just take the bridge and just squeeze it together. Don't worry, the bridge is a little shorter. It's a little less arced than it was at the beginning, but it's fine. Like, people can walk across it, problem solved. Yeah, we lost a little bit, no problem. Yeah, the ends aren't perfectly lined up, no problem. And that would be non-homologous, because homologous means same, so not the same, as in the ends aren't perfectly lined up. Non-homologous end joining. And this leads to loss of info. Loss of DNA. Well, obviously, it's because, imagine the bridge, whatever fell into the water. And that's gone, washed away into the shore. Now, the other one is a good bridge builder. He realizes that some pieces have been lost. He then goes and replicates the pieces that were lost. He remakes them, and after remaking them, he then inserts the he repairs the bridge with the pieces that were lost, maintaining the same integrity, the same arch. The bridge looks like nothing happened. And that is homologous end joining. And there's no DNA loss. DNA loss. Okay? So in this case, we have whatever it was, we just kind of, it's hard to draw it, but let's say the breaks came here, we just like inserted them. Great. Now, let's say that the pieces that were broken are here. What happens here is they remake the pieces, and then our bridge looks beautiful. See how I even drew it arced conceptually? So those are the difference between those two. And in terms of proteins or enzymes, we don't really, I don't think the slides make it clear. If I show you, I'll go down to it. Now, I have to explain the things that's associated with it. I told you, right? I told you very clearly that there's a distinction between things that can be repaired by mutations, mutations that can be repaired by mechanisms, and diseases that are manifest as a result of a lack of repair. Ataxia telangiectasia, or ATM protein kinase. Originally, ATM protein kinase is a protein. ATM protein kinase. This protein, it is like a foramen at the work site. So imagine that there's these people constructing the bridge. There's construction men or women, and then there is a foreman who has to watch, oversee, like an engineer, like a civil engineer at the site. Civil engineer has to make sure things are good before we can move on to the next step. He has to look and say, no, that isn't structurally sound. Let's repair it before we move on. Let's say that there's a mutation or there's a problem that let's say the guy on his way to work, the engineer, doesn't get there. Now the construction workers, they don't care about the integrity of the job. I'm painting them out in a poor light. I'm sure there are construction workers who care about the integrity of the job. But they cut corners. Let's go with that. If the ATM PK, sorry, my throat. If the engineer isn't there, then there's no one to stop the work in the event there's a break. What's going on with my voice? 1 second, guys. I'm gonna go get water. Okay. You'll learn this later. That's why I'm telling you this, because it is of use to you once you get to the cell control lecture. In the cell control lecture, you'll learn about ATMPK. ATMPK is like a civil engineer at the job site who has the job of specifically noticing double-stranded breaks. This will cement the idea home. Its job is to notice these breaks. If the protein isn't working, the kinase isn't working, then there's nothing to stop the cell cycle and say let's not move on to the next step. Our DNA is not repaired properly. There's a break in it. There's no one to do that. So these breaks manifest themselves, continue to occur in different spots in the DNA, and there's no one there to check the system. So you're left with the disease called ataxia telangiectasia. I'm not going to write it out. Ataxia telangiectasia. It is named after the protein that's not working. Does that make sense? So it's kind of like a cruel joke. It's like, imagine there was a bridge that was designed so shitty, and they named it after the engineer who was sick on the day who was supposed to make sure the bridge was built properly. Yeah, that's what it would be like. So this isn't one of those things. Ataxia telangiectasia is not something that can be repaired by double-stranded break repair. It is the case. It is a manifestation of the inability to repair double-stranded breaks, because ataxia telangia ATM, I don't know what the last one is, the M stands for, but ATM protein kinase isn't there or isn't working. Okay? Is that clear? I'm gonna wait for your response because it's a big idea. Great. Great. I'm glad. Do you see this? This is me off Wikipedia. This is me learning a little bit. Ataxia-telangiectasia mutated protein kinase. That's the M, mutated. Normally recruited and activated by the breaking of double-stranded bonds. So when the break comes, that's when normally ATM protein kinase jumps in and goes, hey, there's an issue. But if ATM protein kinase isn't working, nobody jumps in to say there's an issue, so the issue gets to propagate unchecked, and then you're left with a disease. Great. Those are the 5 mutations. I will go through really quickly. That's it. So you've been through the 5. I will go through the difference between loss of function, gain of function, dominant, negative, haplosufficiency, haploinsufficiency. Where is that? Okay. And then after that, we're going to take a little break. So we have haploinsufficiency. You have haplosufficiency. You have loss of function, gain of function. You have autosomal recessive while we're at it, autosomal dominant, dominant negative. Okay, so haplosufficiency, haploinsufficiency are pretty intuitive if you understand the name. Haplo means half, so haplo half. Haplo equals half. Insufficiency equals good enough. You know, when I was a kid, maybe you don't know, but when I was a kid, got tons of S's. Just like satisfactory or sufficient. Same idea. Works well with others. What was it? No, independent work or independent. Yeah, independent work. Always an S. So haplosufficiency means half is good enough. Meaning if you have 2 mutations, 2 alleles, and let's say this one was the bad one, and this one was the good one. This one was the good one. And it was like this, so it was autosomal. The disease was autosomal recessive, right? The disease was autosomal recessive. It has to be, because if it was autosomal dominant, then it wouldn't be haplosufficient. If it was autosomal recessive, then that means that half is good enough to get the job done. Meaning that this one that's bad isn't so bad that it's preventing the other ones, that it's preventing the good from doing what it needs to do. It can do enough. It's like, I don't know, it's like you're having a bad day, you're working at half speed, but half speed is good enough to get the job done. So, no problem. Good enough. That's haplosufficient. Haplosufficient is often linked with autosomal recessive mutations, because if something is autosomal recessive as a mutation, then that means that you need both bad ones for it to be bad, meaning that one good one is haplosufficient. You understand? The sufficiency is discussing is a good thing. It's not like the bad allele is sufficient. One bad allele is sufficient to cause a problem. Don't get it confused. You can just like forget I said that. It is haplo. Oh, I accidentally. Sorry, guys. This one. I apologize if that was confusing you guys. That is haplosufficient. Now, as you would assume, haploinsufficient is the case when half is not enough. So insufficient we're doing now. So now imagine you had a disease that was autosomal dominant. Autosomal dominant means one bad allele is all you need. So if you had this, and I told you the disease is bad enough, and I asked you, did this person be affected? You would say, yeah, affected. Why is he affected? Because this one bad allele is enough to cause a problem, meaning that this one is insufficient. meaning that half is not enough to keep you going good. So if we're going to connect this, I'm going to do this like this with different color, because when I do this, haploinsufficiency is associated with autosomal dominant. And typically, what is the case? I'll explain. Actually, I'll go. Okay, so now dominant negative, the last one you have to go through, and then we'll go through gain of function, loss of function. Because gain of function and loss of function are like a tag on, or another way of describing a mutation. So you can describe a mutation as autosomal dominant, autosomal recessive. You can describe the whole setup as haplosufficient or haploinsufficient, and you can describe the mutation, you can describe the actual mutation on a biochemical level, like on a microscopic, like on the actual physiology, on the level of physiology, as being a gain of function or a loss of function mutation. As in, does this mutation cause something to be overproduced or underproduced? So loss of function is underproduced, and this is overproduced. Overproduced. Wow, I never really used blue. Hating on blue. Underproduced, overproduced. Loss of functions are typically haplosufficient. It's typically the case that if you have one allele and you've kicked it out, one bad allele, and so that it's now working, you've lost some function. It's typically good enough to still run, so it's haplosufficient. But when you have gain of function mutations, like I should know this stuff, but I don't remember because you haven't learned it yet, so you can't really help me here. But it's like oncogenes, or proto-oncogenes, and the RAS gene, for example. There are certain mutations that they cause, even if you just have one mutation, they cause it to overproduce. And these are typically haploinsufficient, because when you start overproducing, it typically becomes a problem. And so they're typically also autosomal dominant, because one bad allele is enough to cause a problem, because you're making more and you can't really balance that out very well. Dominant negative. The difference between dominant negative and haploinsufficiency, or haplo anything, is that in this case, in the case that we've talked, I'm going to clear the screen, because I hope I'm explaining this. I think I'm doing okay. I'll find out. I'll go back to you guys in a sec, and I'll show you. Let's go blue. If you have this case, right, and you have this case, the two the same. This one is autosomal recessive. This one is dominant. Oh, no, let's say autosomal dominant, because that's probably the confusion. Dominant negative. Autosomal dominant. You would, and this was the bad allele. Well, it'd have to be bad, obviously, because it's autosomal dominant. Then you would say this person is running at 50%, and it's not enough. Not enough. So this would be haploinsufficient, because this one isn't enough, so insufficient. This in dominant negative situations, you would say this person is also affected because it's dominant, but they're approximately running at, let's say, 30%. Because this one is dominating, is putting pressure on this one to make even less. So normally it's like there's a bad mutation and a good mutation. The bad mutation, let's say, stops all function. This one's still running at 50%. You would say, and if the 50% is good enough, you would say that's haplosufficient. If the 50% is not good enough, then you would say it's haploinsufficient. If it's haploinsufficient, then this mutation must have been dominant negative. If it's haplosufficient, then this mutation must have been autosomal recessive, because it wasn't enough to cause this to mess up. In dominant negative, once this one is mutated, kind of just like, now it's running at like 20%, and it's also associated with it's not haplosufficient or haploinsufficient, because you're not making half, you're less than half. This one, the mutated one that's dominant, push the production level on the other one down so that it's not enough. Does that make sense? I'm going to pair them up together. I'm going to do this one more time. Haploinsufficient haploinsufficiency is associated with autosomal dominant and typically a gain of functions. A haplosufficiency is associated with autosomal recessive and typically loss of function. Not that this isn't guaranteed. These two are not guaranteed, but it's typically the case. Loss of function, that you can lose half function and still be good. A dominant negative is also an autosomal dominant. It's associated with autosomal dominance because half is one bad allele is enough to cause a problem. Does that make sense? Dominant, when I wrote, oh, sorry. Oh, yeah, that's a good point, guys. Sorry about that. Basically, dominant, when I talk about the percentage, this one is still, when I talk about, sorry about writing in this corner right here. I gotta remember I can't write here. When I'm talking about haplos and stuff, because this is separate than these two. They're still running at 50%. And I mean the good allele. I mean, like, the machine, which is you. You want to be running at 100%. If you have a mutation, sometimes you run a little less than 100. In haploinsufficiency, mutations that result in haploinsufficiency, you're still running at 50%, and the 50% is not enough. Whoa, how did that happen? 50% is not enough. I mean, so you're messed up. So the 50% isn't enough to mitigate the damage of the mutation. That's why it's autosomal dominant. That's why you have one mutation and you have a problem. Haplosufficiency, you're still running at 50% haplo, but it's good enough, and so you don't have a problem. You need both of them to be shut down. You need to have two bad alleles. You need this to have a problem because you need it to go to 0% or a very, very low percentage. Dominant negative is that one of them is messed up, one of them is normal. The messed up one is this one because it's autosomal dominant, but it exerts a pressure so that you're not only, it exerts a pressure on the good allele, so pressure on the good allele, so that the good allele can't run at 50% anymore. The good allele, little a, is now running at, let's say, 30% or 20%. So that it's dominating the little allele, the good allele. The bad allele, so that's big A, dominates little a. Isn't that the case? Poor little a. Bad allele, big A, dominates little a. So even whatever little a was trying to dish out, not working anymore. It's dishing out even less. Does that make sense? Great. And just like that, guys, you have went through all of mutations. Okay? Mutations are done. It's not really that complex when you really understand what we just went through. 5 different types, a bunch of different examples that are associated with types, some being examples that can be healed or repaired, others being examples that are manifest as diseases due to the inability of certain repair mechanisms to function. Okay? I am going to take a little break before I start doing DNA tech. All right, guys. Right now it's 11:20 Eastern Standard Time, so maybe 11:30. I'll start going through this, and we'll see. We'll see how long it takes, and we'll see how we're feeling at the end of this. Okay. All right, guys. I'm going to keep it on, but actually, let me see if I can do something. This might be fun. Come on, countdown timer. Oh. 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 I think I realized it's better to have this. Over here. Hey guys. Chicken. I hope this is working, guys. I hope you are finding this helpful. Just saw a smart car drive by with custom paint job. That's pretty good. Sometimes life just gives you something, you know? Okay. All right. Welcome back. Oh, I have to change this as well. Here we go. By the way, guys, before I start DNA Tech, do any of you have any questions in general about stuff? Don't be embarrassed if you think it's a dumb question. It might be a dumb question, but it's okay to be dumb now, better than being dumb on test day. If any of you have any questions on anything, let me know. You guys are curious about how I went through first semester, what was my study habits, how I reviewed stuff. You guys are a curious bunch. I know I had a bunch of questions when I was going through this process. Okay. No questions? Shall we continue? Previewed hard. I believe it's really important. Okay. Let's continue doing this then. Alright. DNA time. Okay, cool. Maybe at the end. So a lot of people have trouble with this lecture. A lot of people find it a little confusing, a little all over the place. A lot of people have it find it a little bit hard to wrap around because it doesn't seem. Maybe you lose sight of the big picture. Um, the way that Dr. Meisenberg teaches this lecture is that he kind of teaches you basic DNA tech tools, things like restrictive endonucleases, things like denaturing, things like electrophoresis, things like allele-specific oligonucleotides and complementary DNA creation. These are all generic tools. They don't specifically have a function. Well, they don't have a function in and of themselves, I guess. They do, but the function is not typical. Like electrophoresis separates based on size. You know that. But it is used in more elaborate DNA technology that is then when it serves its biggest function, like when you're doing Southern blotting. Southern blotting is a collection of 5 different basic tools that you use at different times using restrictive endonucleases, electrophoresis, PCR, allele-specific ASO, allele-specific oligonucleotide, which is complementary DNA, and probing. All those are the steps of Southern blotting, but they are a collection of all the basic tools. So they will teach you a basic tool, and then they'll kind of teach you an application of the tool in a more complex example of DNA technology. So it's important to not lose sight of that as we go through this process. So these are kind of nothing of use here. First thing is restrictive endonucleases. Restriction endonucleases originally came from bacteria. So I'll give you the rundown. The reason I'm doing this is because I think it's easier to understand than it is to memorize. If you understand things, if you treat it like a story, it helps your narrative really of this whole process, and it makes it easier to understand. So if you have bacteria have things called restrictive endonucleases, and they recognize specific sequences and they cut them. Now, in the event that a virus tries to infect a piece of DNA, this is a virus, tries to infect a bacteria, boom, infected. Here it is. These restrictive endonucleases will scan the virus for any sequences that they recognize. It's specific to each restrictive endonuclease. They have their own sequences that they notice. And if they notice this sequence, they will cut it. And they will cut it such that the virus cannot integrate and replicate inside the DNA. Because the thing about viruses is that the thing about viruses is that they don't have their own replication mechanism. So they need to hijack the replication mechanism of their host in order for them to propagate. So restrictive endonucleases are a set of enzymes, a class of enzymes used that are in bacteria that cleave viruses. Now, the thing about restrictive endonucleases is that they're not specific. They will cleave their own shit if they notice the same sequence on them. So they have something to counteract that. It's basically like, imagine there's a bunch of there's a bunch of roaming police officers. The city's on curfew, and they're roaming the streets to make sure that there is no uproar, no violence, no resistance starting, things like that. And they look, if you aren't wearing a patch, I'll say you have to wear a patch like that. If you aren't wearing a patch that lets you know that you're cool with the government, that you're a law-abiding, friendly citizen, then you will be dealt with. They're gonna go, hey, who's not wearing a patch? If you're not wearing a patch, I'm gonna cut you. It's a little aggressive of the government, but the concept is the same. So DNA of the bacteria have their own way to put on these patches, and these patches are just methyls. They just methylate themselves using methyltransferase. The name is very obvious. It's very intuitive. The name is transfer methyls. So they'll transfer this methyl onto their own DNA to prevent them from getting in trouble with the roaming police, the roaming militia, the roaming guards, whatever it is. But the viruses, they don't know this. They don't have the opportunity to put on their patch. They can't trick you into thinking that they're a member of your country or city, so they get dealt with by the restrictive endonucleases. So that's basically the original discovery of restrictive endonucleases. We then found a way to make use of them. Now, I hope that's clear. That's the history behind restrictive endonucleases. So there are two types of ways that it can cut. So here's a double-stranded DNA. You can have sticky ends, which just cut right down the middle of a sequence. So say here's a sequence and here's the complementary sequence. It'll cut right down the middle of it, leaving you with blunt ends. These are called blunt ends because they're blunt. Okay, blunt ends. Or you can have things that cut. So let's say you have our sequence. You can have things that cut at different spots along the two sequences, along the strands, and that's left with sticky ends. So you're going to be left with something like this, and something like this. Okay, it's like Tetris. These are called sticky ends. They're of more use to us. These are of no use to us scientifically. No use, yeah, because there's no way we can insert our DNA into this, because it will just like fall through conceptually. There's no overhang. But in this, we can insert our DNA right here, and it'll stick, basically, because this will have something to stick to, and this will have something to stick to. It's a little, the drawing's not the best, but the point is there. Now, we realized, we being scientists and humans, figured out a way to utilize restrictive endonucleases and plasmids as a means of replicating our own DNA. As a tangent, though, I want you to make the distinction between a palindrome. I've done this to my tutoring class, the people that come to my session. I make the distinction between a palindrome in English and a palindrome in DNA. So if you have like A, G, C, T, T, and you have A, G, G, A. This right here, the bottom one, this is a palindrome in English. And a palindrome, because I think they actually gave us a question on our mini, basically just said identify which of the following sequences is a palindrome. And they will always have this as a way of tricking you, because they have to make sure that you actually know the difference. Palindrome in English is same forwards as it is backwards. So you're thinking this, if I were to read it this way and read it this way, would be the same. Right? So you go, okay, palindrome, wrong. You'd be wrong. What makes a palindrome in DNA is if the 5' to 3' of the original is the same as the 5' to 3' of the complement. So if I drew the complement out, 5' to 3' would be G, C, T, T. Do you see how this is exactly the same as this? This makes this a DNA palindrome. And restrictive endonucleases, REs cut at palindromes. These type of palindromes. Oh, sorry. These types of palindromes. Okay, distinction clear? Don't get this wrong, it's free marks. Just know that if it looks like an English palindrome, chances are it's not. It can't be. If it is an English palindrome, it cannot be a DNA palindrome. Because if it's an English palindrome, that means that the ends are the same, like A, and that means that the reverse will always be T, or whatever it is, G's. It'll never be the same. For it to be a palindrome, conceptually, if you don't want to write out the other thing, this has to be the complement of this, this has to be the complement of this, this has to be the complement of this. But that might not be simpler for you. Just draw it out 5' to 3'. That's clear? Yeah, it gets people. I know. Does that make sense to people now, though? Palindrome distinction made? Um, Other DNAs. So, great, cool. Now, how do we use it? So this is called recombinant DNA technology. I would write it out, but I'm just gonna write oh, I hate when this happens. My mouse is getting weird. Touchscreen. Recombinant DNA. That's gonna be annoying. Technology. Hopefully it fixes itself. Recombinant DNA technology. Okay, cool. Basically we have our DNA. We want to use it at the end of the day. So we have our DNA. And then we have a synthetically made plasmid. Plasmid has a bunch of stuff on it. It has 1, has 2. I should change the colors. It has 1, has 2, and it has, let's see, 3. 3 things that are of importance. First thing we do is we take a restrictive endonuclease. Example, ECO R1. You don't have to know where they cut. You don't have to know the sequences. You don't have to really know much. But let's say you take ECO R1 and you cut this, and you're left with some sticky ends, because we said that sticky ends are the only ones that are of use to us. Make it a little more. Okay. So you're left with some sticky ends. I should have drawn it in red. This is red. The black thing is in red. Then we take the same thing and we cut this. Now, if you look in this, there are sites for cutting in one of these sections, ECO R1. There's a bunch of different sites. This right here, if I'm not mistaken, is the lac Z gene. Right? This right here, that's not the right color. This right here is the ampicillin resistance gene. Now, I'll explain it to you conceptually first, and then we can start getting into the details. So basically, what's our bigger goal? Why do we use recombinant DNA technology? The reason we use recombinant DNA technology is we want to take our DNA and replicate it in a cost-effective manner. Now, the easiest thing that we can conceptualize to replicate DNA is to insert our DNA into a bacteria, because bacteria grow a lot faster than we do. They replicate the DNA a lot easier. Is to insert our DNA into a plasmid, which is a circular DNA basically. Ours is artificially produced. Insert that into a bacteria, and then watch it grow. And then when it replicates, the hope is that it replicates ours too. But there's a bunch of things we have to do to make sure that that our DNA. So there's 2 things that we have to make sure of. We have to make sure, 1, that our DNA, by our DNA I mean the DNA that we want to replicate, which is this one, is in plasmid. Okay, first thing. Second thing we have to make sure is our plasmid is in the bacteria. That's it. And that's what these 2, this green box here and this yellow box here, I don't want to change the color, but that's what this and this check. This one checks that, this checks 2. And this checks 1. Okay? So the lac Z gene is used to ensure that our DNA is in the plasmid. The ampicillin gene, ampicillin resistant gene, is to ensure that our plasmid is in the bacteria. And if all those things come true, if 1 is true and 2 is true, then we should be able to ensure that we've replicated our DNA and we've extracted our DNA from the bacteria that has correctly replicated our DNA. It's a lot. But, so, if you know what each gene, if you know what each thing does, if you know what the LAC-Z gene does, and you know what the ampicillin resistance gene does, then it'll make sense too. It'll help you make sense of it. So the LAC-Z gene, if you were to take this plasmid with nothing inserted into it, so this gene is intact, this gene is complete and untouched. If you were to go and place this into a bacteria, so I'm going to go like this, into a bacteria. If you were to insert this into a bacteria, what would happen? Well, this gene would be synthesized, right? And then it eventually make a protein, right? And that protein would basically make the entire bacteria blue. Which means that if you looked at a petri dish and you saw blue bacteria, you would know that this plasmid was correctly taken up by the bacteria, and then this plasmid was synthesized or replicated, and the protein was made, and we were left with the blueness. So if you see blue bacteria, you know that this plasmid is in it. If you would see white, or I think well, if you don't see blue, so if there's no blue, oh, you can't read this part. Sorry. I realize that you can't see this section, so I gotta make sure. Blue. Sorry about that. Habit. Oh shit. Okay. So this makes it blue. Now, in the event that restrictive endonucleases, I want to insert my gene. Into my plasmid. So what I do is I cut it with the same restrictive enzyme, like an ECO, as I cut here. So what this does is open up right here and then insert my gene into it. So if I were to draw a new one, the thing is there's not a lot of room to draw a new one, but I'll try it anyway. So if I were to draw a new one, let's say I draw it inside it, it would be like this. Then you'd have a little bit, then you'd have red because my gene has now been inserted. And then there would be a little bit of yellow here, a little bit of yellow here because we've cut in the middle of the yellow and placed our gene in. What that does is prevent this gene, this yellow gene, the LAC-Z gene, from being synthesized. So what that does is prevent the bacteria from being blue, right? We said blue was associated with the yellow gene. When the yellow gene is cut in the middle, that means that the gene doesn't get to get synthesized completely. It's like you took a protein and cut it in the middle and then put shit in between it. You took a DNA sequence, cut it, and put stuff in between it. There's no way that that DNA sequence will create the same protein anymore. So you won't get that blue making protein. And if you don't get that blue making protein, then the whole bacteria isn't blue. So I told you how this checks one. If we have a blue protein, then we know our DNA is not in it because the yellow gene, the LAC-Z gene, was able to be made cleanly. If it isn't blue, so if it's white, so I think the other color, if it ends up being white or not blue in general, then you know that our DNA must have been correctly inserted into it, preventing it from being blue. Does that make sense? Yeah, I'm gonna have to, because it's important to understand why you have those two things in there. Okay, I'll try to explain it one more time. Eat some chicken. Where is it? Sorry. One more time. You guys have a picture of this, or are following this? Now, you have, I'll explain it one more time. You have a yellow gene. This yellow gene So we have a blue and we have a yellow. This yellow gene makes bacteria blue. So if you were to take this and insert it into a bacteria, and if the bacteria had properly taken up this, then when the bacteria goes to replicate its DNA, it's going to replicate this obviously because it's inside it too, and it'll make the bacteria blue. Don't know why, I don't understand it, but it's blue. So now when I look at a petri dish full of a bunch of bacteria, some of them are blue, some of them aren't. I know the ones that are blue correctly have this plasmid in it. Right? So all of them should more or less be blue. If I've done nothing, if I've correctly taken it up this plasmid. But I now have my DNA and I've cut it such that my DNA is inserted in the middle of this gene, right somewhere in the middle of it. So we've discontinued the yellow gene. We've made it two parts. Now the yellow gene can't make its protein anymore. So it can't become blue. So when I look at a petri dish and there's white stuff, I know the white stuff must not be making this gene, because if they did, it'd be blue. So that's. Hey guys, sorry about that. Are we still there? Hello guys, are we okay? Okay. Yeah, my program just crashed for a second. Okay. So that is what does the first thing. That is what checks that our DNA is in the plasmid. The second one, I know it's a little complex, guys. It's a little tricky. The second one is the ampicillin resistant gene. So how that works is, after we want to make sure that when we extract the bacteria, we extract only the ones that have our plasmid in it. So this plasmid makes this bacteria resistant to ampicillin. This will make a protein. This gene will make a protein. I don't want to write like that. Resistant. This will make a protein that makes you resistant to ampicillin. So when I have successfully inserted my DNA into the plasmid, and then I will flood all the bacteria with ampicillin. Most of them will die, or the ones that will die, do not have this gene being made. And if they're not making this gene, then that means they haven't correctly taken up the plasmid. So only the ones that live have our plasmid in the bacteria. And only the ones that live that aren't blue have our DNA in the plasmid and have been taken up by the bacteria. So we want not blue, plus alive. If those two criteria are met, if they're alive, that means they have our plasmid. If they're not blue, then that means they have our DNA in our plasmid in the bacteria. That's the best I can do, I think. I hope that helps. I hope that makes sense. So if I show you here, this is what I described. This makes you resistant to antibiotics. Awesome. This makes you resistant to antibiotics. This makes you blue. If we cut it and insert our gene, great. I don't want to run so our DNA and a human plasmid must be in bacteria. Yes, in order to replicate it, you need the DNA of interest that we want to replicate inserted into the plasmid. It's an artificial plasmid, so it's not necessarily human plasmid, it's just an artificial plasmid. We need our DNA in the plasmid, and then we need the plasmid taken up by the bacteria. If those two things are met, then we will have, if the LAC-Z gene does not contain an insert, the colony is blue. If the LAC-Z gene is disrupted by an insert, the colony is white. Great. And this is exactly what, one more time, fragment vector, recombinant vector. Recombinant vector into bacteria. Boom, recombinant vector now in bacteria. Antibiotic, antibiotic. Ones that have ours, alive, replicating. Ones that don't have it, dying. Done? You guys understand restrictive endonucleases? Done? That's it. Okay. Great. Now, complementary DNA. Complementary DNA is taking RNA and mature RNA and reverse transcribing it into DNA. Where am I? Here. So now we're going to go through complementary DNA. So, complementary DNA involves taking a mature. Involves taking a mature messenger RNA and bringing it through reverse transcriptase to complementary DNA. Still single-stranded because this was single-stranded. And if it was double-stranded, it wouldn't be very functional to us. We want it to be single-stranded so then we can then try to hybridize it to DNA. So for this to make sense, you just need to know what is in a mature mRNA. So if you know what's in a mature mRNA, you have the cap. You have the cap, 5' cap. That's a methylguanine, so it's good to know that there's a methyl G. You have the poly A, 3'. Sorry. You have a 3' poly A tail. You have your 5' untranslated region and your 3' untranslated region. And then you have exons. So this is everything that you have. 1, 2, 3, 4, 5. 5 things that are in a mature mRNA. Then you take those and you make them into complementary DNA. So you change the U's to T's and so on and so forth. Now the thing about complementary DNA is they no longer have introns in them. So when you get that question that discusses and also, make sure you remember that this stuff is in a mature mRNA. This stuff is what has promoters and enhancers and upstream signaling things. So things to basically help everything get set up. It doesn't get made into amino acids because the methionine and the stop codon are before it. Are like so it's like 5'. It goes cap. I guess I'll just clear it. It goes cap. Then you have 5' untranslated region. Then you have exon, which has a methyl. Methionine. So met. Then you have a stop. Then you have a 3' untranslated region. And then you have a poly A tail. So those are the components of mature mRNA. So by making this into complementary DNA, there's no introns, which is functional because it allows us to hybridize with only the things of the DNA that will actually go on to make proteins in the body, because so much of your DNA is junk. You only have 1.3% of all your DNA is coding exons. So imagine that's 98.7% of your DNA does not make proteins that are useful to you. So it's very important to be able to find where those 1.3% are and what they do, basically. So you use complementary DNA. You take a mature mRNA, you make it into a complementary DNA, and then you go hybridize it to things. And it has so many different functions. Remember how I was telling you that you learn basic tools first? This is a basic tool. This isn't more than that. And the best way that you can kind of understand it is to do that question. You know the question that had this? As soon as I start drawing it, you guys know what I'm talking about. To this. And then it goes from this to this to you can't see, but I made it trail off again. I guess I'll try to do it one more time. Sorry. I hope that still works. It trails off because this is your poly A tail, poly A, which has nothing to bind to because there's no poly A tail in the coding in the original DNA sequence. And it asks you what happened. And so you might think that there's, because they tell you this is DNA and this is RNA. This is a side note, but if I help explain this, so this is complementary. Well, it's actually complementary DNA, but it originally was RNA, right? It was originally mRNA, but it's the same thing. So you go, what happened? Well, I'll explain. First of all, you have to understand, what are these? What are those? So these are introns to B. I know it doesn't make the most, never been told this, but DNA doesn't have introns. DNA has coding and things that will eventually be non-coding. But when they took the DNA and they created an RNA, you had introns that were then cleaved out because a mature mRNA doesn't have introns. So why these loops exist is when I hybridized the complementary DNA made from the mature RNA back to the original DNA strand, the introns to B have nothing to bind to. So that's why they're forming these loops, because there's nothing for them to bind to, because that part is was cut out in the process of making the mature RNA. So now you look at this question right here, the one that's below, you look at this situation, and you go, well, what's happening? I only see one intron to B, and there used to be two. So you might think, well, oh, they asked you what happened in this individual, and the question, so you might think, oh, there was a mutation that cut out the DNA. You can't have giant chunks of DNA removed. So it's not a viable option to think that the DNA was removed. Even though this is gone, it's not gone. Let me show you where it is, and this might make sense to you. It's right here. That's why it's longer. In the question, they don't draw it longer, which is why you might find it confusing. But it's right there because a splice site in the mRNA was removed. So it didn't have the opportunity to splice out that intron. So that intron was mutated, that mature RNA was mutated, and it kept that intron. So when I made the complementary DNA, that intron is now able to bind to that intron to B before, because it was never removed. So it flattened it out, removing one of the loops. And that's what happened in that question. It's an abolishing of an intron splice site. Does that make sense to people? I'm going to wait a little bit because that's an important thing, because it helps you understand the parts of a mature mRNA and what isn't there. Okay, great. Awesome. I'm glad. So that intron is gone. And that's why it has nothing to bind to. But in the event that the intron was not gone, it does have something to bind to, the intron to B. And the intron to B now binds to the intron, which is right here. This red part was an intron that never got removed. Now, yeah, because you can imagine if you remove this, this would loop up, and the whole thing would get shorter, because this part would connect to this part. Okay. Okay, guys, complementary DNA. You understand the parts of it, you understand generally its function. You use it to then identify things in the original DNA strand. So we can figure out where the exons are, where the coding region is, that kind of thing. Now, that is just the general use of cloning DNA. Now they tell you, what do you use? See, I told you at the beginning, they give you a generic tool. So we have seen restrictive endonucleases, generic tool, which then got to be complicated or utilized, be made more complex through the use of recombinant DNA technology. So restrictive endonucleases, recombinant DNA technology, the advanced use of this technique. Next one, complementary DNA. These are the advanced uses. So you can see cloning, you can see gene expression, because if you can know where the exons are, then you can know the gene expression, because the ones that become exons are expressed. And then you can use it for hybridization, which is the whole idea that you want to bind it back up to a DNA and see where things are. Now, they're going to explain what hybridization is. Hybridization is really simple. The idea that we have a piece of complementary DNA, and we have a piece of actual DNA. You don't really get to see this. You don't get to see this in and of itself. You don't just get to see DNA. You have to you have to basically fluorescently label things and then notice things. So imagine this is a coding region, right? Coding. You know it's coding because this is exons. So what they do is they probe it with a fluorescent probe. Okay. And then they go and they see where it binds. Where is it going to bind on this thing? We don't know where, and we don't know this. Like, we don't know this thing, this big blue thing. We don't really know where it is or what things are on it. We just know that you have DNA for the purpose of my explanation. Now, eventually, let's say it binds here. Oh, sorry. Let's say it binds here. Okay? It binds there, and here are your probes. Now we know that this region, let's say this was an allele for mutation. And this is in RNA, remember. Now we know in this location on the DNA is the DNA allele for, oh, you can't see this, sorry. It was allele for mutation. Allele for mutation. Now in this, we see that this is the DNA allele for the same mutation because it hybridized, it binded to this region, and I have these probes that let me know where it is. So I found it. If I didn't hybridize it, if these weren't there, if this wasn't there, then I would have no idea that it binded here. It wouldn't be of use to me. But because I have these little green fluorescent probes, I am then able to identify where it binded. So now there are things that you can do with this. And this is the cool thing, right? This is a little more of a, we're taking hybridization and we're making it, we're upping the tool's use. So now you can make a synthetic probe. I can make a probe. And I can label it with fluorescent labels. And let's say this is a synthetic probe because I made it. But I already know through other studies previously, I already have this information. I already know that there is a certain sequence that seems to be associated with sickle cell mutations. So I make this sequence. Here it is. It's this thing. I made it. This is the sequence that if you have it, you have sickle cell. Great. Now I want to know what you have. So you originally have two pieces of DNA, right? We have a DNA that's double-stranded. I separate them using heat, because you know that, and I create this, and I create this, right? That's why you have two alleles. No, that's not why you have two alleles. You have a piece of DNA that I separate, but you have two alleles nonetheless. That's here. I'll do it better. You have allele 1, and you have, okay, we'll go like this. And you have allele 2, right? Mom and dad gave you both these alleles. I separate them using heat. I separate both of them using heat. And then I'm left with one allele of one allele of the other, and one allele of one allele of the other. And I put them on separate sides, and I see where the synthetic one chooses to go. Obviously, I have to have 2 of these because I have to make 2 choices. If, let's say, one goes here and one goes here, and let's say this is the mutated and this is the normal, that means that I have one mutated allele and one regular allele. Based on, it's like, I like the metaphor I like to use, it's like putting a dog between 2 parents who had, like, let's say a couple had a fight and they're separating. And you want to know who the dog loves, who the dog is going to go with. You put them at opposite ends of the hallway, and you put the dog in the middle, and you say, choose dog. And the dog goes to the owner that most complements it. If the dog, let's say you are a mutated dog, and you prefer the mutated owner. You guys get along. You're both messed up. Let's say you had a depressed dog, and you had a depressed owner, and they both get along because they both are wallowed together. Okay? Then the dog gets to go and join that owner. Let's say you have a really high energy dog and they always like to go on walks, but only one of your owners likes to really go on walks. The dog will prefer the one that goes on walks because it most complements the dog's makeup. Does that make sense? And that's what ASO is. This is allele specific oligonucleotide. So I made an allele specific to sickle cell. So that's the ASO. It's an oligonucleotide because it's from 5 to 10 or 10 to 12. Oligo means more than 1, but not 2, more than 3. Like oligosaccharides, a bunch of sugars. Oligonucleotides, a bunch of nucleotides. And then this is called blotting. So you probed it because you have to know where it is. And then you put it in the hallway and you said choose. If they both go to the normal versions, then you know that you're homozygous normal. You're homo-normal. If they both go to the mutated one, then you're homo mutated. What's it called? You're homozygous affected. I don't know what the word is, but you're homozygous for the mutated allele. That is how allele-specific nucleotides work. Does that make sense? And that's called blotting. Any questions? Any questions so far? If there's any questions, let me know. If not, I'll continue. Okay. And that's what this represents. This represents, they say A is normal, because you can be HbA, hemoglobin A, and S is hemoglobin sickle, HbS. That's messed up. Now you look, you gotta check where the dogs went. There's 2 dogs, 2 owners, 2 dogs. So both dogs can go to each owner. Okay? So one allele bind to the normal, one allele bind to the sickle. This person has both. This person has both. 3 and 5 are heterozygous, meaning they have each allele. They possess each allele, meaning one of the dogs wants to go to the normal one of the dogs wants to go to the messed up one. So that makeup is one normal, one messed up. Number 6, both of them went to the messed up one. Number 2, both of them went to the messed up one. So the 2 and 6 are homozygous affected. Nice, I got it right, homozygous affected. And then 1 and 4 are homozygous normal because both the alleles are on the normal. It's really simple when you kind of understand what's going on. Okay? Great. Fish. This is still using hybridization. This is still using probing. But instead of using very small specific sequences, which the purpose of ASO and blotting was to let you know what allele you specifically have, or which alleles you specifically have, because you have two alleles. The point of fish is not to tell you what alleles you specifically have. It lets you know larger information about what regions of chromosomes you have. Like if you know a certain mutation is associated with an inversion or associated with a translocation from chromosome 6 to chromosome 7, and if you have that translocation, then you have that disease. So what you will do is you take that large region, you probe it, complementary DNA, you probe it, and then you see where it goes. If it goes to 6, then it's normal. If it binds to the 7 in the messed up chromosome, if it binds to a different chromosome than it's supposed to, then you know that that's associated with the mutation. So it just lets you know where certain things are on a macro scale. It's not specific to the sequence. It's not about a sequence. It's about general, large chromosomal sequences, large chromosomal sections, and whether or not you have certain translocations, certain mutations, like a trisomy, if you have a duplication of a certain segment of your DNA, things like that. So it's much more broad. Okay? Understand fish, understand ASO. Now electrophoresis. Electrophoresis is just separation based on size. The bigger things get stuck, the small things move faster. So you cut them up to a bunch of pieces. What have we already learned that allows you to cut things into a bunch of pieces? We've already learned a way to cut things. So what is the way that we have learned to cut things already? Oh. Anybody? How have we learned to cut things already? Good. Yep. Let me get back to it. Exactly. So it's another, see how it's little tools being used. So once you cut, now that you understand electrophoresis separates based on size, we're going to go into southern blotting. See, southern blotting isn't anything new. Someone wrote snowdrop. In case you guys haven't seen it, this will just be a quick way to help you remember. Snowdrop. Snowdrop. Looking on. So there's southern blotting, northern blotting, western blotting. Southern blotting is associated with DNA. Northern blotting is associated with RNA. And western blotting is associated with proteins. You shouldn't get it wrong now. That's it. That helps for that. Now, there are 5 steps to southern blotting. If you understand what southern blotting is, what the purposes of southern blotting is, ultimately, the ultimate purpose of southern blotting, then you can understand why and what the steps are. Don't just memorize the steps, because you can't just say, okay, like I can look at this slide and I can go, oh, okay, digestion, separation, denaturation, blotting, probing. I can go DSDBP, DSBPP, DSDBP, and then I got to remember them on the test, and then I got to remember, okay, those are the sequences, and then maybe they don't ask you that, maybe they don't ask you the sequence, maybe they ask you what tool you would use in each step, or maybe they ask you what the step really means, or what's the next step, and they describe an experiment that they've already done and say, what's the next step? And so you should know where they are in the process. You should really understand the process. So at the end of it, if you understand the main purpose of southern blotting, because southern blotting will be a question for sure on your test. It was on mine, it was on my midterm, it was on my mini 1, and it was on my final. So southern blotting, but if you know the ultimate purpose, then it's okay. The purpose of southern blotting is to basically figure out whether or not you have a specific allele of interest, whether or not you have a specific allele of interest, to find it without necessarily knowing where it is to start. So you're starting from kind of square one, but you want to know whether or not you carry the sickle cell gene, but you don't know it yet. You don't know you don't know where it is, you don't know, but the sickle cell we do know, but if it was something that you didn't know where it was, but you knew that this was the specific, if you had like a certain sequence, I don't know, GGCG, whatever it is, let's say it was GGCG. If you had this sequence, then you were mutated, or at least you had a mutated allele. Mutated. So Southern blotting needs to take your DNA and needs to take you from just a big piece of DNA to something that is able to hybridize to something small to really get to the process. So the first thing you have to do, imagine I gave you a handful of DNA. It's a long strand of DNA. You can't do anything with it yet. It's just your DNA. It's just really long, and there's 1,000 bases, 2,000, 100 kilobases, whatever it is. First thing you want to do is break it down. So that would be digestion. And what does digestion use? How do we cut down big DNAs into smaller pieces of DNA? Restrictive endonucleases. Great. Now I have a container. Okay, I have a container full of pieces of DNA, different lengths, based on whether or not there was a cutting site. What now? I don't need all the different lengths. Let's say I know that my sequence exists, my mutated allele sequence, this one right here, exists between the 5 base to 10 base length. I know that the mutation is 5 bases, 10 bases long. I have all these different pieces collected in a container, all these different lengths. That's of no use to me. I need to find a way to get just this length out. So what's the next step? Well, you would think, okay, how do I separate it based on size? Okay, so that's separation. And how will we learn to separate based on size? Electrophoresis. Electrophoresis. Now that we have a bunch, now I can isolate. Okay, I look on the gel here. Okay, and there's all the different sizes, and I go, these ones, anyone in this region, is between 5 and 10 base pairs. That's good. Let's extract them. Great, now I have them. Boom. Now I have a collection of just 5 to 10. That's good. Now what? These are still double-stranded, are they not? These are originally double-stranded. I cut them. They're just a bunch of double-stranded in different lengths. You know, they're still double-stranded. I need to eventually take my allele-specific oligonucleotide and my complementary DNA and hybridize it to the original DNA. So what do I have to do to the couple, to the double-stranded DNA? I have to separate them into single-stranded pieces, or else there's no way. I need to make the dog choose eventually, right? That's the goal, is to find out where the parents are and find out which parents the dog chooses. It's like it's kind of like that. You have like 1,000 couples, and a dog, and 2 dogs. And you need to know whether these dogs, which specific person these dogs belong to. So first thing you do is you take these people and you break them off into smaller groups. So you break them off into couples, let's say. So rather than have 1,000 couples, you have, rather than have just a giant collection of people, you now have 1,000 couples. Okay, great. Now I have to separate the couples. So that after I have 1,000, so now I have to. So this is when I took a big group of people and I made them into couples, and then I picked the couples that were only. I made them into groups, and I picked the couples that were only 2 people big. That's that. That's these 2. Now I take this couple and I have to separate them. So I take this double-stranded DNA, which is small, right? 5 to 10, which let's say that equals 1 couple. Doesn't matter. And then I have to separate it into this and this. This and this. Why is that working that way? Great. What's that process called? That's called denaturing. Because it's unnatural to break up a couple. Denaturing. Unnatural. Denaturing. And that's done using heat. You've learned that already. Heat. So now I'm going to erase, so Southern blotting. First one we said was digestion. Cut them down into small pieces. Right? Make them manageable. Separate them based on the pieces that I want, size-wise. Denature denature them so that now they're single-stranded. and then I blot them. Is it blot and probe or probe and blot? Blot then probe. Yep. So I blot them because now that we have separate them, you blot them. What does it mean to blot them? It means you take the couple right here and you put one here, and you take the other one and you put one here. So you put them on either end of the hallway, right? So imagine you have a hallway, and then you put the dog. Good enough. And you gotta have two dogs because each person has two dogs, two alleles. And then you see which way they go. And that fifth step is probing. You took this, these dogs are colored, so I see them, and then I go and see which way they go. And based on where I go, I now know which sequence you have. If they went to each, then you're heterozygous. If they went both to one, then you're homozygous. If they went both to the other, you're other homozygous. And that right there is southern blotting. How does that work for you guys? Does that make sense? Does that make sense, guys? That works? You guys still with me? Everyone still following along? Should I call it? It's 12:30. I don't like I'm still good. Like I don't have much to do. I can do this. I don't have much to do. I can do this for till we finish all of DNA tech, but you guys have been at this for 2.5 hours. You guys still want to continue? Okay. All right, let's get through it. Okay. So southern blotting is done. Now, so this is another basic tool using restrictive endonucleases. So we're using restrictive endonucleases which cleave at certain spots, right? Now, every person is different in terms of where their cleavage site is and whether or not they have polymorphisms surrounding that cleavage site. Some people will have different fragment lengths. Now, whether or not you have different fragment lengths means that when I probe for specific regions, I will see different region lengths based on your probe, based on where you cleave. I will see different fragment lengths based on where you cleave, which is, so I have a probe that checks this region, okay? Now this person has cleavage sites here, here, and here, or this allele has 3 cleavage sites. This allele has only 2 cleavage sites, meaning that the length of this one is 1.15 and the length of this one is 1.35. So when the probe comes, the probe is only able to see the region that is attached to it. So the probe, for example, this, if you have HbA, the probe, you'll never be able to see this region because this region doesn't have a probe lining up with it. So the first thing you would do when you attack these problems is look where the probe lines up with the question. So where the probe lines up with the DNA strand. So in the event of, if you have something like this, so imagine you have 3 possibilities. You have something that's like this, and I'll draw the cleavage length, the cleavage sites in a different color. You have this and this. you have just this, you have this, and this. Now, in terms of where the probe is, let's say the probe is right here in length. So you would go, okay, this is 1 kilobase. Say this is 1 kilobase, and this is 5 kilobase. That means this must be 1 kilobase, this must be 1 kilobase, this must be 6 kilobase, this must be 4 kilobases, and this must be 2 kilobases. This is just numbers that you'd be given in a question. Now, these are 3 different alleles that you can have. Oh, you can't see my hands. These are 3 different alleles that you can have. So, if you had allele A, what lengths would you see on your electrophoresis? What length bases would you have? If you're following along, then you would see 5 and 1. In this one, you would just see 6, and in this one, you would see 4 and 2. Now, why that is because look where the probe lines up. The probe lines up. So in this one, the probe touches this region and this region. So you get to see these two. The probe touches this region. So you get to see this 1, 6 base. The probe touches this region and this region. So you get to see these two. You'll never get to see this. All of them have this. It's not like they don't have it. If they have, let's say, if you had any of these alleles, you would have a segment, a restrictive fragment length polymorphism that is 1 kilobase, 1,000 bases. I will just never be able to identify it because my probe doesn't hybridize to that region. Doesn't mean you don't have it, you still have it. So based on what fragment lengths you have, I can tell what alleles you have. So if somebody had fragment length 5, 1, 5, and 6, what would they have to be? What would their allelic makeup have to be? So you think, 5, 1, he has a fragment length that's 5, he has a fragment length that's 1, he has a fragment length that's 6, he has to be AB. He has to have this allele and this allele. Because how else would he have these lengths? So this guy would be AB. If you had just 4 and 2, what would you have to be? You'd have to be CC. Because you don't have 6 and you don't have 5 and 1. That makes sense? So that's why, look at this individual. He has 1, like this AS, AA, SS, this business won't be given to you on a test. This is what you're supposed to figure out. So if you have 1.35 and 1.15, then you know that you have this length and this length, so you must have, okay, no problem. Basically, the probe hybridizes to a certain region. So this probe is here. This probe then hybridizes to this region, or any of these regions along the length of the probe, these regions right here. You have different fragment lengths. You have different fragments. You have a 4 and a 2. You have a 6, and you have a 5 and a 1. Now, the probe will notice any region that hybridizes to the probe. So this 1 kilobase hybridizes to the probe. Even if not all of it, it still hybridizes. This 5 hybridizes to the probe. Maybe not all of it, but it doesn't matter. It's bound to that sequence. So you notice that sequence. It's the same thing when we did those questions where we had things like this, or when I was talking about complementary RNA, and it was like this. And I told you that now we know this whole section. Now we know it because it's bound to it, so we can see it. Okay? Same concept. So, based on the fact that every allele has different fragment lengths because there's polymorphism in an individual, you have different spots where you can cut because you have different restrictive enzyme sites. So, based on the different lengths that are noticed in the ASO, in the probing process, I'm able to know what allele you possess. Because if a person, Where is this? I have to, just because there's no way, it's getting messy. If a person has here we go, 3 possibilities, A, B, C. Whoa, that's the worst. A, B, C. So we said that the probe notices this region. Whoa, slow down, baby. Probe notices this region. Okay, great. That means that it notices from here to here, from here to here, from here to here. That's what it'll see. Okay? Now every person has different spots that they cut. Let's say you cut here, and you cut, I'll do better. Let's say you cut here, and let's say for some reason they all have the same cutting spot. But now this person has one cut here. This person has one cut, no cut. This person has a cut here. Now you look and you say which regions, first one, which regions in the A1 are lined up, is possessed within these lines. You know, this sequence is, and this sequence is. This has 5, this is 5,000 bases, this is 1,000 bases. You look at this one, in this region right here, from this line to this line in B, you know that this whole region will be noticed. There's no other cut spots, so all you get 6,000 bases. In this whole region you have here will be noticed, and here will be noticed. This one is 4,000, and this one's 2,000 bases. So it'll notice those. So if somebody came in and you did a test and you checked which ones their probe hybridized to, and they had 6, all you saw was 2 sets of 6,000 base pair sequences. You would go, okay, well they only have the 6 length. That means they must have this allele. Whoa. That was anticlimactic. Hold on. That means that they must have this allele times 2, because they just have 6. They only have, all I can see is a fragment length that's 6,000 bases long. This person, if I saw someone who had 5 and 1, that's what I said, if someone had a length that's 5,000 bases and a length that's 1,000 bases, and I saw a length that's 6,000 bases, so I know that if they have 5 and 1, they must have allele A. That's why they're A, because there's no other way you can get bases that are that long out of these 3 alleles. And if they have 6,000 bases, the only way you get a 6,000 base is if you have this restrictive fragment length polymorphism. So you must have allele B. So they must be A and B. They must be A and B. If this person has 4 and 2, you don't see anything else, then they must have C twice, because everybody has 2 alleles. So they must be CC. And if I take you to this idea, this person is doubled up on 1.35. They don't have a length different than 1.35. So you look here and go, they must only have this allele twice. So they must be homozygous mutation. Both of their alleles are the mutated one. That's why they're this. If they had 1 and 1, then they would be heterozygous, and that's why none of these people have mutations. None of these people are actually, look like this person's dark black because they're mutated. And this one is not mutated because they're heterozygous. And to have sickle cell, it's a autosomal recessive. You have to have both. Does that make sense? And if you guys have the DNA technology questions from Dr. Meisenberg, there are some good ones that really help you go through this problem. If you guys actually post the DNA technology questions that you got from Meisenberg in here. And if you post them, then I can go through one of the questions that'll help you make sure you don't make the mistake that other people make. I'll go on until you guys post. Until you post the DNA tech thing, I'll move forward. But you're always going to get a question on this. yeah. This isn't that big of a deal. Tracker and link markers, not that big a deal at all. Like it's just something to memorize. It's not not gonna get asked on it. Basically, the idea is that in family studies, inheritance of a mutation can be tracked using a polymorphic site. So, mutated genes missing one cleavage site that is recognized by the probe. See, fragment detected by probe, phenomenon detected by probe. This one is the wild type has this cleavage site. The mutated one is missing that cleavage site, and so by hybridizing to it, you are able to then you're able to track the mutated allele. But like this is another example, same idea. Mutated allele has caused a different number of repeats in the mutated allele, so in the different number of repeats, you can hybridize to that, and that'll allow you to distinguish. It's just basically different ways to separate wild type from mutated alleles and track them. It's like different techniques that you can hybridize to. Different things that are unique to individuals that you can hybridize to allow you to track certain alleles in individuals. I hope that makes sense. It's just not terribly important, and there's better ways to spend your time. You guys have the DNA technology questions? I don't know if you can post. I guess maybe you can't post them. Can you not post a link to? Maybe I'll go through it. I just know that it's pretty valuable to you guys to be able to do these restriction fragment length polymorphism questions, because you will guaranteed get a question on it. And just certain topics you know that are such high yield. Look at how much you went over it in your class and how much it kept on coming up. It's okay. I'll go through the process like this. Thank you, Janelle, but I'll just go through it and find it this way. It's this week 2 stuff. DNA tech, DNA questions. Okay, very easy so far. So probes here. So what sizes would you see? This is like a really simple one. If this probe is here, what sizes would you notice? You would notice size 6, and you would notice size 4. You would not notice size 2. You wouldn't notice size 2 because the probe doesn't line up with it. So in the question on what you would notice 4 and 6. Simple enough. Good. Good, guys. So easy enough. Another question. This is just conceptual. You should be able to look at this and say, what's the makeup of all these people? And this is for the cystic fibrosis gene. So this person, both dogs went to 1. Both dogs went to the mutated. Sorry, both dogs went to normal, both dogs went to mutation. They split up. Your alleles split. One allele was normal, one allele was mutated. Your alleles didn't split. So you're looking at the makeup of these people, you would go normal, affected, heterozygous, normal. Normal, affected, heterozygous, normal. Great. Now these are the type of questions I get people. Let's see if I can do I got an idea. Let's see. Okay. No luck. Really? Let's discern it. I'm gonna go with copy, paint, open, paste. Okay, great, that's better. So in this situation, okay, they're gonna ask you what's happening right here. That's what this question comes down to. What's happening here? Okay. Now look, there's a common mistake that you will make here. You'll make this mistake all the time. Here's the mistake. I've been through this with the guys who came, with the people who came to my tutoring session before the hurricane. There's a common mistake that people make in this process, and I'll show you it. So here it is. Look, you're gonna look at this, right? You know, this first person, this grandpa, 1-1, mutated. So you're gonna go, okay, he has 5.7. So let's say first thing you do is you want to break down these into alleles. So what I do is I say A, B, so much simpler, C. Okay, A, B, and C, 3 possible alleles. If you have these 2 lengths, because the probe is right here, if you have these 2 lengths, you have A. If you have this length, you have B. If you have this length, you have C. Great. So this first person has 5.7, has 3.2, and 2.5. What is this person? Well, you would look, 5.7, 3.2, 2.5. This person must be A, C. Right? You look at mama. Mama is doubling down on 3.2 and 2.5. So doubling down on 3.2, 2.5. So mama is AA. And here's where you'll get a mistake. I know it. Look, you guys, if you've made this mistake, then you know where I'm going. You're gonna look and say this person is mutated. This person isn't. So C must be the mutated one. Great. C is mutated. You're feeling good. You're gonna come down to the bottom ones and go, okay, C. This person has C. This person has C, because these are the 3 kids. They're lining up. Oh, but by the way, if I didn't tell you, they all line up. So you can take a line down, and they line up to their situation. And this is always the case. Always the case. So you're going to go, okay, this person has it, this person has it, this person has it. So these 2 have, this guy's doubling down on it. So this person must be homozygous, mutated. This one must be hetero. And this one must be hetero. And that would be your answer. And I bet you that's one of the choices. Homozygous, heterozygous, heterozygous. And you'd be so wrong. That's not the way this question works. You don't get to do that. It's too simple. You don't get to just look at what the allele that the father has and then say it's mutated, the grandfather, and that means that anyone who has that mutation has it. Let me make that very clear. You cannot just take the mutated allele that the grandpa has, which is mutated. You're right. You're right about the fact that this is messed up. But you can't just assume that every person who has it, of the kids, has it. Because look at this woman right here. She married into the family. She's not of the family. She has the same allele. But she's fine. Meaning that you can have a length that's this long without it necessarily being a mutation. It's not like when 2 people, like let's say I have a heterozygous recessive disease. I'm going to say that one more time before I jump on. You can have the same fragment length as someone with a mutated fragment length without having the mutation. So it's not like in cases where someone is where a disease is autosomal recessive, meaning both parents have to provide the messed up allele. They don't provide the same messed up allele. They're not family. They don't carry the same messed up allele. They carry different messed up alleles. So don't think that that the only way to get this mutation is to have a messed up 5.7. You could have a messed up something else hypothetically, and then get the allele, and then end up being mutated. So, going back through this process, you're right so far. We're right that this is mutated. Now, you look at this kid, this one right here. He carries his dad's mutation. So he's C. Now what I like to do, because like I said, you have to do this. It's a messed up C, so I mark it like that. And it's a messed up C, so I mark it like that. This person is what? They are C, and then they have 3.2 and 2.5. 3.2 and 2.5 is A. So this person is CA. They must have got A from mom, and they must have got the C from dad. And they're mutated. Great. Now their partner is what? this woman right here, she is 5.7 length and 4.7 length. 5.7 and 5.7 and 4.7. So she must be BC. BC. Okay? But her C is normal. Right. It's the important distinction. It's what'll always be the case in every question you get. They will always put this in there to really distinguish that you understand how these alleles work. That you're not just like, because they have to make sure you truly understand it. They're not trying to trick you. I mean, it is the trick to the question, but if you did not have this additional factor into the question, then you'd never be able to distinguish whether or not you truly understand the concept. So she has C, but she's normal. So now you look at her kids. This kid, so if I'm gonna go, I'm gonna transfer this over here. AC, CC, no, sorry, BC, that's the wife. This one is messed up. Okay. Now let us look at their kids. Their kids right here are, this kid is, first one is CC, 5.7, 5.7 right here. This one is CA, and this one is CB. Okay, so CA, so C with everything else. So let me put that in. CC, CA, and CB. Great. Now look, I don't even need the numbers. That's what makes this beautiful. Mama gave this C, this B, right? That's pretty simple. You can start with ones that you know. Mama gave this B. So this C must have come from who? Must have come from Dad, right? So if Dad brought this C, he also brought the disease C. Now, you look at this. This is why you might get the question wrong. This A must have come from Dad, right? Mom can't give an A. There's no A's. No A. So Mom can't give this A. So that means that this C must have come from who? This C must have come from Mom. And it's normal. It's not mutated. Yeah, it's 5.7, but it's not mutated. Now the CC, this one's really easy, the last one, or the first one, depending on how we're going. This C goes here, and it's mutated, so we're going to make sure because Dad had to have given a messed up C. And Mom must have given a normal C. And if Mom's given a normal C, then this one, so now who has the mutation? And it's autosomal dominant. We obviously saw that because when anyone had a mutation, they had the disease. They were black squares. So first child is mutated. Second child is normal. Third child is mutated. This is different than the original answer that we got. The original answer we got had them being heterozygous, homozygous mutated, heterozygous mutated, heterozygous mutated. That had 3 people messed up. Either way, they were all mutated. That's different than the case with the question. So now if I go back to this, it's saying which individuals in generation 3 have received the mutation. Now we know it's only 1 and 3 because this C is Mama's C, not Dad's C. So the answer is B, 1 and 3. Very different than the answer you would have gotten if you just went from the top down. If you would have skipped some steps and not thought about the fact that she can have the same fragment length and not have the disease. So the answer is B in this question. How does that work out for you guys? You see a very important distinction to make. Great. I'm glad. Good. PCR. I hope that you guys have went over PCR in the past. It's not bad. You have to also make, this is important, to know the distinction between when you use PCR and when you use recombinant DNA technology. Remember we talked about using the vectors at the beginning to replicate your DNA? When you have very little amounts of DNA, when you have, it's precious, you can't afford to be putting it into a bacteria and some take it up, some don't, some die, some don't. It's too much cost, too much risk when you don't have a lot of DNA. You use PCR. Use PCR when you don't have a lot. So the example is pre-implementation, pathogenic DNA, crime scene DNA. All these examples are you don't have a lot and you can't afford to waste it. I am losing steam, so I'm going to call it right now, and then maybe later tonight I will pop back on and finish the rest of the DNA technology ones, because really there's not much. It's not to the same capacity as the questions that I went through. Like, they're a lot more serious than what I had went through was a lot more serious than this stuff. This stuff is like, it's conceptual. But I appreciate that. Thank you. I will get back to it another time, and maybe I'll just post the end of it for you guys. Also, non-homologous end joining and homologous end joining, you've already went through this, so this is similar. You went through this in mutations, so it's kind of interesting. You know this. No problem at all, guys. I'm going to end the no problem. I'm going to end the slide. I'm going to end the thing right now. If you guys have any questions, feel free to message me or post it in the group. If there's any ways you think I can improve this, or what the case is, I'll let you guys know. I might be switching over to Twitch, which is a different streaming process. But also, yeah, we'll see. I'll let you guys know. No problem at all, guys. Okay. How do I close this? Give me a second. Have a good day, guys. Do practice questions. Also enjoy time with your family. Don't stress out. Have a good one. Peace. Peace.